excuse me. Hello everyone and welcome to NOAA's Omics Seminar Series. My name is Tracy Gill and along with Catherine Egan, we are the webinar co-hosts for this series. Before we get started, here are a few logistics, also listed in the chat box. For this webinar, we have two presentations and the presenters will address your questions and comments at the end of each presentation. All attendees are muted, but feel free to type your questions and comments into the chat box at any time. If you want to go to full screen or a larger view, find the 4-arrow button to the upper right of the slide deck. Hit that, and this 4-arrow button toggles the full screen view on and off. If you are interested in getting a copy of the recording or a PDF of the slides, we will send them a day or two after the talk to all who registered. If you are not on the weekly NOAA Science Seminar email list, but you would like to be, please contact me at tracy.gill@noaa.gov and I will add you to the list. And I want to thank our captioner for making this webinar more accessible. Thanks. And now I will pass the mic to my co-host, Catherine Egan, who will introduce the NOAA Omics series and today's topics and presenters. Catherine?
All right. Thank you so much, Tracy. And hello, everyone. Thank you all so much for joining us today. Welcome to the NOAA Omics Seminar Series. As Tracy stated, I'm your co-host, Catherine Egan. I am the OAR, Omics Coordinator for the Oceans Portfolio, and I also sit in NOAA Ocean Exploration. I also sit on the NOAA Omics Working Group as a subcommittee co-lead. The Omics Seminar Series is hosted by two NOAA groups, NOAA Ocean Exploration and the National Ocean Service, through the One NOAA Science Seminar Series. Tracy Gill and I will be co-hosting the Omics seminars, and as she said, I will be moderating the questions today. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. So with that, we have two omics presentations today from two scientists with NOAA's Atlantic Oceanographic and Meteorological Laboratory, or AOML. We'll have our first speaker present, and then we will take questions after this presentation, and then we will begin the second presentation at around 12:30 p.m. Eastern Time. The first presentation is titled The Use of Omics in Environmental Forecasting, presented by Dr. Anderson Mayfield. Dr. Mayfield is a coral reef researcher from Nashville, Tennessee, who has been developing omics methodologies for corals since his time as a PhD with the late Dr. Bruce Gates. Upon coming to NOAA in mid-2019, he has been attempting to use omics and advanced big data analytics to develop diagnostic tools for reef coral health assessment. Thanks so much for presenting today, Anderson, and I will now pass it off to you. Anderson, if you are talking, we cannot hear you right now.
Is that any better?
Yes. Perfect. Start again. Thanks.
Oh, that's strange. Okay, well, anyway, sorry. Well, hey, everybody. Thanks for joining in. I'm Anderson Mayfield, working at NOAA's AOML Coral Program. Today, over the next 20 minutes or so, my goal is to kind of give you a little taste of what we're doing at AOML in terms of using omics and environmental forecasting. But I think you'll quickly see, because of the nature of this seminar series as being the NOAA Omics Seminar Series, I'm going to dedicate much more time than I normally would to the molecular end of things. So I'm actually interested in all facets of coral biology, from the ecosystem scale down to the colony, down to the polyps, and then into the cells and molecules. And I'm particularly interested in developing diagnostic tools for coral reef management. And I'm hoping you'll see in the first few slides that I'm going to try to sell to you why omics might play a role, and for one interested in doing these kinds of things. So I'm going to spend a little bit of time on how I think omics could serve to be a tool in ecosystem forecasting. And even though I basically exclusively work on coral, I've tried to make this talk more general in terms of the guidelines, recommendations, and whatnot. So although the data I'm going to show are from coral, most of the images are going to be from coral, I want to definitely promote this as something that others of you out there that might be working on, fish or microbes or something else, this could be advantageous for your own research. And I've provided Tracy with a much more detailed version of this talk with 40 or 50 slides, because obviously I'm not going to be able to go into too much detail, but I want to make sure people that do want to consider using proteomics in particular as a tool have this information. But you're of course also more than welcome to email me about stuff. So really the reason why I think omics could play a role in health monitoring for coral and other marine organisms is this idea that subcellular biology should reflect aberrant behavior before you see these visible, more extreme, late-stage manifestations of health decline. And I think I always use the analogy of the heart attack. We're going to the doctor annually or ever so often to have things like your blood pressure measured, your biomarker panel assessed, because we know those things like blood pressure and cholesterol levels that are going to be diagnostic of later declines in your physiological condition. So omics has the potential to give us this kind of information for marine organisms. Is there a gene or a metabolite or a protein whose concentration or activity could tell us something about later decreases in resilience to, for instance, climate change stressors, which is particularly important for coral. So I've been using a lot of omics data in building predictive models for forecasting resilience. And I'm not going to talk too much about the data analysis side today because it's been a topic of other seminars I've given at NOAA, but I definitely feel free to reach out to me if you want to get more details about that. I do want to briefly say that it's not exclusively omics. So if you want to characterize the resilience of an ecosystem, you do want to have this health informative data from the omics. But that doesn't mean you're ever not going to want to do these more tried and true survey measures, looking at seawater quality, looking at ecological factors like organismal abundance and diversity. So it's not to say that we'll ever, for most projects, the best case scenario is you're going to have data across multiple scales of biological organizations. So you're going to have the omics scale data, physiological scale data, ecological data, and all the way up to the seawater quality. That's going to allow you to build a more powerful predictive model in which you could assess an animal or a habitat's resilience. So although the remainder of my talk is going to be exclusively about omics, I do want you to keep it in the back of your mind that we're not doing omics at AOML in isolation. We're factoring in these ecological and environmental data as well. And I'm going to give another longer talk about this in a few weeks if anybody's interested. So the last few years I radically changed my research from doing almost exclusively gene expression based stuff to what we call proteomics. And my definition of this is essentially it's just the assemblage of all proteins in a sample. This could be a cell, a tissue. Really the technical definition is it should be all proteins potentially synthesized by an organism. What we use a little bit more colloquially is just a collection of proteins. So if you remember your basic molecular biology, you've got your central dogma in which a DNA is transcribed into a messenger RNA, which is then exported from the nucleus and translated into a protein in the cytoplasm. And I encourage you to stick around after my talk. You're going to hear a really excellent seminar on coral gene expression from Evelina Rubin about some of our work in the Port of Miami. I gravitated from genes to proteins just because I think these proteins are actually undertaking the work within the cells. If you want to address fundamental issues in cellular behavior, such as things we study at AOML, like how do animals respond to stress, which proteins are involved in climate resilience, in my mind it makes sense to look at the proteins because these are actually modulating the behavior in cells. So that's So the mechanistic side of things I'm still very interested in. But as per the last few slides, I think this idea of making health conjectures from proteomic data and this biomarker capacity is kind of the other reason why I'm interested in proteins. So that's more of the applied side of things. I apologize for the kind of chaotic nature of this table, but I do want to take a minute to go through it, and I made this specifically for the people listening who already have an omics background. I tried to make this talk a little bit more general to not be off-putting to non-molecular people, but I did also include some things that are, I'm directly speaking to other people doing omics. And I think this is the kind of thing I would have liked to have known years ago when I first started doing proteomics and nobody told me. So now I'm doing you guys a solid by letting you know this. So how this table works, don't worry, we're not going to go through the whole thing. You've got your macromolecules we regularly profile on the left, your DNAs, RNAs, proteins. Lipids are technically metabolites, but I've separated them here. We've got our jargon for what we call the technology, the chemistries, but what I really want you to focus on is this. So first of all, you can see right now the price per sample of doing omics is in that $100 to $200 ballpark, and it doesn't actually differ that much across these different omics. Some people get better prices than others, and amplicon sequencing might be cheaper than this, but you can see it's within an order of magnitude. It's actually relatively close. What's very different is the data output. So right now, I could teach a high school student to get good enough quality DNA to sequence the genome, and assuming, and same goes for RNA. You realistically, with as good as next generation sequencing technologies are these days, you should realistically be getting 100% coverage of whatever you extracted. If you didn't extract everything, of course, you may not get the whole genome, but suffice to say, whatever you ultimately purified should be getting sequenced. On the other side of the spectrum, lipidomes, for instance, you can quantify a couple of hundred lipid species. There's over 100,000 different lipids in a eukaryotic cell. You're getting less than a fraction of a percent by profiling a couple of hundred. Doesn't mean you shouldn't do it, but it means the coverage of the lipidome using current technologies is really low. Proteomics, so what I'm doing is squarely kind of in the middle. So you could get 20, 30, 40,000 proteins in an experiment from the mass spectrometer. But in reality, the amount that are going to end up in your paper, as you'll see, ends up being much lower. And there's a variety of reasons for this. There's methodological reasons, there's bioinformatic reasons. And again, I want to kind of shed some light on this for people, not to put you off from doing proteomics, but just to make sure you have more realistic expectations when you embark on these. So you're probably only going to get 1 to 5% of the proteome using current technologies. So even though I'm actually doing proteomics on a day-to-day basis, I still extract everything. So we really shine in doing multi-omics extractions at AOML. I've been working with this Caribbean coral, Orbicella faveolata, and we extract the RNA, DNA, protein, lipids, and metabolites. My particular interest right now is in proteins, but I want to have these RNAs, DNAs, and other molecules in my back pocket. Maybe my data lead me to believe that there's a microbial shift in my sample. I can go back to my DNA in the freezer to look into that. So although I'm only going to be showing proteomic data today, I did want to emphasize that we have good techniques for doing these multi-omics kind of extractions and analyses. And I would hazard a guess that most people listening are probably more, if you are familiar with molecular techniques at all, you're probably more familiar with DNA and RNA stuff. So I've kind of developed these slides accordingly. But all these omics protocols go through the same basic steps. You're preserving the samples the same way. You're snap freezing in liquid nitrogen, putting them in a preservation buffer. You're extracting the molecules, so you're lysing cells. You're purifying the different macromolecules from one another, so the RNAs go here, the DNAs go here, etc. then you need to put these molecules back into solution typically so they can be analyzed. So they need to be dissolved or eluted. The proteins, lipids, and metabolites need to be separated to some extent before sequencing. And then the hallmark of all these omics technologies is being that, you might spend days to weeks on the extractions and laboratory analysis, and you're going to be spending months to years trying to make sense of the data. So the bioinformatics bottleneck, even for these smaller lipidomics and proteomics data sets, the bioinformatics step is always going to be the bottleneck. That's something in common with all these approaches. So here's kind of, I envision this as being like, I was sent out into the world to learn all this stuff about proteomics, and I took back the knowledge I gained to share things with you guys that I think are important for framing work you might want to do. And this is like the proteomics tip number 1 that's kind of the elephant in the room, and that's you're never going to dissolve all these proteins you extract. Protein biochemists have known this for years. This is not something truly new that I uncovered, but it's not something that people really want to deal with in manuscripts because it might cast doubt on your data quality. And coming from a gene expression background, this is very different, because if you have a DNA pellet at the bottom of a tube and you coat that with tris buffer, you realistically should be dissolving everything that's there. You might lose a little bit, but it's trivial. Similarly, if you have RNA on a spin column and you add some DEPC treated water to it, you should be eluting all that RNA. This is not the case with proteins. You could use the strongest anionic detergents on the planet, like sodium dodecyl sulfate. You could add denaturants like urea. You'll eventually solubilize a good portion of your proteins, but then you get to what I call the proteomics catch-22. All these detergents and denaturants that you need to use to dissolve your proteins, you need to remove them before you do your omics. And then a huge chunk of your proteins are going to come out of solution. That's a big problem. Nobody ever talks about it, even though it's been an issue for decades. So this is one of the reasons why the proteomic data sets tend to be smaller than the nucleic acids. So for the proteins that you are able to dissolve, the quantification step is easy. You're using assays like Bradford and Lowry that have been around for decades. You basically use standards. Most proteomics people don't run gels, and I think this is a big problem because you do want to know something about the quality of your sample. The problem is, to get a gel that looks like this is actually the nicest gel I've ever run, and you can see it still looks kind of blurry. This took 100 micrograms of protein, and you might only extract that much from a small sample. So thankfully, the mass spectrometry core that I use at the University of Miami, they have a nanoliter scale gel system, and yes, the gel looks way crappier, but you could still see some protein bands, particularly in the rightmost column. So really what you what you don't want to see is, I thought it was going to come up there, you don't want to see a glob here at the bottom of the gel. Same for RNA and DNA work. You might have a great concentration on the nanodrop. You go to run those nucleic acids out on a gel, it's totally degraded. You basically want to make sure that's not the case. Once you have your proteins quantified and you're okay with the quality, you've basically got two choices. In both cases, you digest these proteins up with enzymes like typically trypsin, and then you can either directly inject these trypsinized peptides into a liquid chromatography instrument, which I call shotgun proteomics, and then you proceed to literally just sequence whatever was in your sample. You get presence-absence data, so it's semi-quantitative at best. What might be more attractive to many people is to do what's much more analogous to RNA-seq, in which you basically digest up your proteins, stick little tags on them, and quantify the concentration of those tags. Very analogous to tag-seq. You can do 8 to 20 proteins in a run, depending on whether you use iTrack, which is the approach I use, or Thermo Fisher's tandem mass tag. So you might say, hey, given similar cost and similar protein needed and whatnot, why would you ever not want to do the fully quantitative way? And this is because only 5 to 10 or maybe 15% of the peptides actually get the label. Yet another thing nobody ever told me, it's not in the manuscripts. Fact of the matter is, most people are going to be totally oblivious to this unless you really look behind the curtains of the bioinformatics. So this leads to high batch-to-batch variation. You have a protein that's labeled in batch A and not batch B. So at the end of the day, this is cutting into the ultimate number of proteins that are going to be publishable in your manuscript. So right here, I've done both of these methods, and we're talking about several hundred proteins that were basically quantifiable at the end of the day. So regardless of whether you label the proteins or not, you're going to treat them the same. You're going to run them through a liquid chromatography column that separates them. They get sprayed into a mass spectrometer. The one they use at the Miller School of Medicine in Miami is the best on the market. It's designed by a friend of mine. It's the Q Exactive. It's one of the most sensitive analytical instruments ever developed. Bashes apart your peptides, analyzes them in a special mass analyzer, and you get these really esoteric mass to charge ratio profiles. So basically, if you do the label-based method, the first pass through the instrument gives you the peptide sequence, which you can see at the top here. The second pass gives you the label intensity of your label. So then that's the proxy for the concentration of that protein. And it will work very similarly for the competing technology, TMT. The problem is, this figure kind of lulls you into complacency that there's a nice, neat relationship between this mass to charge plot and the peptide sequence, and in reality, it's a lot of smoke and mirrors. The freeware available is terrible. I try a different one every month. They all require pre-processing on commercial software. They have bugs. The commercial software tends to come with the instruments, and they're $20,000 a license. Basically means you're dependent on your core facility to use their license. So because the UM facility uses a Q Exactive, which is from Thermo, I use Thermo's Proteome Discover, but it's a black box. You take the data from the instrument, you compare it against your genome or transcriptome, and it spits you out an answer. You have no idea what's going into that. So, and I do want to re-emphasize here that de novo proteomics is not there yet. You need to do some preliminary DNA or RNA sequencing first if you want to have a hope of identifying these proteins. This will certainly change, but right now you need to have some nucleic acids data first. Thankfully, even though it's all smoke and mirrors, what you actually get from Proteome Discover in this case is a pretty easy to read table. You've got over here, you've got your peptide sequences, the modification, so you want to make sure it has that iTrack label. Remember, most of them won't. This is a particularly good example. And these are the two values you really want to gravitate towards, the PEP score and the Q value. This is more, if you're familiar with the E value for nucleic acids research, it's kind of the same idea. It's a probability score. So you want these values to be approaching 0, because high values, basically, this is the probability that this sequence is incorrect. So in this case, it's a 100% chance this sequence is incorrect. I don't even know why it bothered to give me those data. So this would be filtered out. Q value is essentially just the false discovery rate adjusted version. So you want these to essentially be approaching 0. And this will, so given kind of some of these things I've mentioned, this is how you can go from 40,000 peptides I sequenced from the mass spectrometer down to, in this example with Orbicella, I'm only going to publish or talk about 86 in my manuscript. Q value, so mapping issues because the peptides are so short, iTrack label issues, batch variation. These are the elephant in the room issues that you're not going to see in people's manuscripts because it's not really going to be the kind of thing you'd want to highlight. But I'm actually okay with these numbers. 86 proteins gives me enough to understand changes that are happening in my corals in response to temperature, It's not going to be a fully characterized proteome, but it allows me to do some hypothesis testing. It allows me to build AI-based predictive models in which I can forecast coral resilience. So the data sets are smaller than I'm used to from a gene expression background, but I'm still getting enough proteomic data to be able to resolve differences in coral that could be useful for what I call coral reef triage. So basically, I just want to reiterate that yes, these data sets are smaller given current technological issues that I've reiterated over here, but I still think proteomics could play a role in basic research of coral, other marine organisms, and could be useful for environmental forecasting. And I look forward to kind of running with this idea of not just proteomics but other omics into incorporating them into this predictive modeling framework in which we could use omics and AI, in my case, in transitioning it into a decision-making tool. This could be useful not just for this figure where I'm monitoring coral health in situ, but also things like identifying resilient genotypes, identifying optimal restoration sites, mitigating environmental success, and things like that. So I think, as long as you're cognizant of these limitations relative to nucleic acids research in particular, I think you could still hit the ground running and have proteomics advance your research immensely. And I look forward to more people in the NOAA sphere incorporating proteomics in the future. So with that, I apologize I went a little bit over, but hopefully there's still time for some questions, and I'd be happy to take any. Thank you.
Thank you so much, Anderson. You did not go over, and we do have a couple minutes for questions. It looks like we have one from Tracy to start. Have you been able to help NOAA managers with any coral issues yet using these techniques?
Basically, this is such a new idea that we're still at the stage of, well, the fact of the matter is we need to publish our first big paper on it, and we had some COVID-related setbacks because I wanted to get more field validation data. So I think that's kind of the first step, is get this big paper out on using omics for resilience forecasting, and then we already have a good relationship with Florida Keys. National Marine Sanctuaries will be a logical place to start. So we already have some identified stakeholders. So I think once we kind of put the finishing touches on this, we're hooked up with all the right people, all the right managers to really realize this or transition this. And I think it's not as far-fetched as it might have been 10 years ago when I pitched something like this. I really do think that we're nearing a point where we could, distill the data into a little bit more user-friendly format and give it to a manager and say, hey, based on our omics data, reef A is 2 times more resilient than reef B. Do with that information what you will.
Thanks, Anderson. And I think this maybe answered Tracy's second question, which is about a situation where you'd help a coral manager. So it sounds like what you said, you use omics to provide them with information about which reefs are resilient or not. Does anyone else have other questions for Anderson? We have another minute or so. Looks like we have some folk typing in the chat, so just wait one moment.
No problem.
That. All right, looks like we have a question from Kaylin. How can coral managers in vulnerable communities use omics? Are there differences in cost and tools accessible?
Yeah, this is always a good question that comes up, and it kind of gets back to one of my early slides. Let's see, I can just go back. Because it bothers me to say that, okay, I might have the world's best coral stress test, And it would bother me, though, if I went out there to, I used to work in the Philippines and Indonesia, I'm not going to expect that they're going to be able to go out there and do $50,000 worth of omics data. You know, this is not, the money's not there, the cost bothers me. So what I actually tend to do, and this is another reason which I didn't mention, why I look at a lot of different factors, because I want to make this cheaper. I don't want to say that anybody who wants to measure the healthier reef needs to do $10,000 worth of omics. So I look at all these relationships among other types of data with my coral health data to try to see if there's something cheaper or maybe a smaller subset of things we could be measuring, because I do think it's unrealistic to go, even in the U.S., to go tell a marine park, hey, you guys need to do $50,000 worth of omics a year to assess the healthier reefs. This bothers me, and I'm trying to figure out ways to make it cheaper. I tried to incorporate some of the proteins into a microfluidic chip that you could take on a boat, but that ran into some other issues that I won't go into. But yeah, I want to make this a more egalitarian approach that's, you know, it's not only going to benefit the U.S., but I haven't figured out how I'm going to do this yet. The price point bothers me, and I think that's one of the reasons why managing coral reefs based on temperature alone, which has been done for decades, is attractive, because there's a very nice tight relationship between temperature trends and coral health. So I just need to figure out how to make this cheaper and how to get buy-in and show that it has value in addition to these other, you know, in addition to or alongside these cheaper, more traditional reef management tools.
Excellent. Thank you so much, Anderson. So we're at time for Anderson's presentation. However, if anyone else still has questions, I really encourage you to please type them in the chat, and we can continue to address those after our next presentation. So thanks again, Anderson. And with that, we'll move on to our second presentation now. This presentation is titled Molecular Mechanisms of Coral Persistence within Urbanized Locations in the Port of Miami, presented by Dr. Evelina Rubin. So Dr. Rubin received her MS degree in 2009 from Nova Southeastern University in Florida, where she studied stony coral recruitment to ship grounding sites. In addition, she received her PhD from Stony Brook University in 2014, where she studied molecular mechanisms of pathogenic virulence and host resistance to disease. She gained additional molecular biology and next-generation high-throughput sequencing, as well as bioinformatics experience during her postdoctoral studies at the University of Connecticut in 2014 to 2016, and at the University of Rhode Island from 2016 to 2019. She joined the Accrete research team at NOAA in November of 2019. She is using RNA-seq methodology to understand transcriptomic responses of corals surviving in marginal reef environments and to provide an insight into molecular mechanisms that enable their resilience. So thank you, Evelina, for presenting today. I will now pass it off to you.
Okay, hi everyone. Can you hear me? I'm just going to check first.
We can hear you.
Okay, great. So thank you so much for attending this seminar. Like Tracy said, my name is Evelina Rubin, and I'm a research scientist at the Collaborative Institute of Marine and Atmospheric Sciences, which is a joint institution between NOAA and the University of Miami. And I will be presenting results from our study focused on measuring transcriptomic responses of corals that live in highly urbanized waters of Port of Miami. And our manuscript on the study just got published like about a month ago. So if you get interested after this talk and want to learn more and read the whole publication, there is the link for the publication. So there you have a picture. There's a picture of Port of Miami, and the waters here are not pristine, oligotrophic waters that you think of when you think of a coral reef. They are greenish with a lot of plankton and turbidity. And besides these poor water quality condition, we find these beautiful corals that are on the second pictures that look super healthy and well. While the corals of the same species on the offshore reefs are usually diseased or bleaching or almost entirely vanished from the reef. So we asked this question, what is different? Why these corals in Port of Miami are doing so well? And can we use transcriptomics to learn anything about molecular mechanisms contributing to the persistence of these urban corals? So this slide shows the location of our study sites. First, of course, you have the map of Florida highlighting the location of Miami, and then we zoom in into Port of Miami, where you have 3 urban coral sites. We had MacArthur Causeway North, MacArthur Causeway South, and Star Island. That's those 3 little dots, and there was a pointer here that we could use right there. And then we have our control site. That is a nice reef here offshore, about 6 kilometers of the Key Biscayne. That is, by the way, a regular traditional reef. So I just want to highlight again the major location differences between the Port of Miami sites and the more pristine Emerald Reef site. The urban sites are shallower, about 1 to 3 meters deep, compared to the offshore reef, which is deeper, located at about 6 to 9 meters of water. And this has, of course, implications in the amount of light that these corals receive, so for photosynthesis and energy. The shallow depth creates more rapid temperature and pH changes. We do measure temperature in these different sites, and the water temperature at urban sites are colder in winter and warmer in summer, so the variability is super high. Because of the urban sites are part of the Biscayne Bay, they are more eutrophic with frequent plankton blooms and crashes. Also, the nearshore location causes frequent stormwater runoff, which leads to increased bacterial loads. Okay, and so this is the whole omics design, and I want my talk is not focused on omics as much, and that's as much omics technology I will have for the whole presentation. It's just a little slide. So we collected 31 samples from 31 corals. So from the urban corals we collected 10, 6, and 9, and from the Emerald Reef we collected 6. It's a very interesting story because we dove Emerald Reef at least 3 to 4 times to find surviving Pseudodichloria strigosa corals, and it's just very difficult to find. They're almost non-existent because of the recent disease outbreaks. But after we have the tissue, I isolated RNA, and then I sent it actually my total RNA to sequencing center, where they can prepare the whole RNA-seq or transcriptomics. They do cDNA amplification and library preparation, sequencing. These are Illumina, different Illumina machines. That's what they look like. And then I receive billions of sequences, 30 to 40 million reads per sample, where they have to go through very complicated pipeline to get some meaningful results. And I have listed some of the bioinformatics packages for those of you who use transcriptomics under that little computer screen, and you may recognize them. These are for many different types of steps during the bioinformatics analysis. And jumping straight to results, so we have some results. So in addition to doing transcriptomics, we actually estimated symbiont composition. So we found that all colonies from the Emerald Reef, which is the offshore reef, were hosting predominantly symbionts from the genus Braviolam. And then all colonies from the MacArthur North side, which is one of the urban sites, so this is the shallowest and the furthest away from any tidal exchange, contain only Durasinium symbionts. And then the Star Island and MacArthur South had corals that had a kind of different corals that had either Braviolam dominated or Durasinium dominated. And we're thinking this is because Star Island and MacArthur South are more exposed to tidal exchange and still are receiving the Braviolam symbionts that are originally always described to be the primary symbionts for this coral. This result is important because the Durasinium symbionts are more heat tolerant and stress tolerant, and they are frequently hosted by corals in very turbid and not in warm waters. So we already learned from this first result that one mechanism of corals to adjust to these urban conditions was to host more heat stress resistant symbionts. This is a principal component analysis showing the similarity among the 31 coral colonies. And the way this is constructed is just basically transcriptome-wide expression profile. So I use expression of something around 39,000 transcripts to show the similarity between samples. And the coral colonies cluster based on location and based on dominant symbiont type within the coral. So specifically the corals from Emerald and MacArthur North, and these are the blue dots are the Emerald Reef, and the red diamonds are from the MacArthur North, have very distinct transcriptome expression profiles. And then the corals from the Star Island and the MacArthur South, which are the green triangles and the yellow squares, you can see they kind of were located in between the two sides. So they were having kind of not as high differences, they were kind of falling in between. But then when we labeled the same PCA with the dominant symbiont type, we got very nice clustering showing us with tightly cluster of corals that hosted Braviolam and then the ones that hosted Durasinium. And since we observed both the effect of location and symbiont type on corals' transcriptomes expression profiles, we did multiple differential gene expression comparisons. And again, the gene expression comparison is just analysis for us to see which genes or which transcripts contribute the most to the differences between our different coral groups. And then to reveal the effect of location, we compared the gene expression of corals from the urban sites, which are listed here, and we compared each one to the corals from the Emerald Reef, since that's our control site. And then we also did comparison for gene expression between Braviolam-dominated and Durasinium-dominated corals. So altogether, from the 4 differential gene expression analysis that I mentioned in a previous slide, we had 2763 differential expressed transcripts, so around 3,000 transcripts that were different between these different comparisons. And in order to make some kind of understanding what's happening, you have to first assign a function to your transcript, which is part of that bioinformatics pipeline, and then possibly one way of doing an analysis would be to group the transcripts into broader categories, such as here the broader categories could be metabolism or gene information processing or particle recognition. You can see the gray area is the unannotated transcript. So we still have many times a lot of sequences for which we don't know the function of. And that's very normal for many organisms and even for humans, since we really lacking many functional genomic studies to understand what the genes are coding for and what they for, but that's quite abnormal. And then we looked out of all of these different groups. I selected 4 that were the most interesting and had the most interesting patterns showing the differences between the urban and the offshore corals. And these were immune and defense response, genes, digestive and degradation enzymes, all proteins related to transport, intracellular and transmembrane transport, and then particle recognition receptors. So the top tiny bar graph that you can see shows the number of overexpressed, the dark blue, and underexpressed light blue transcript in urban corals in comparison to the offshore corals. So, the end graph only includes transcripts including particle recognition receptors. So these are all transcripts, and here's the number of these transcripts, but these were all categorized into the particle recognition receptor, and we can see how many they were differentially expressed between these corals. On the left, you see the full list, kind of even further categorizing the particle recognition receptors into further different categories, which I'm not going to, of course, bore you with. I'm just going to highlight as an example. For example, lectins are now molecules involved in food particle selection and in sensing pathogenic organisms. The overexpression of these molecules in urban corals is the most likely response to the higher number of particles present in the water where they live. These receptors are most likely assisting urban corals in heterotrophic feeding, for sensing prey, and help them discern potential pathogenic organisms. In addition, lectins may also assist these corals in detecting symbionts and aid in symbiont shuffling. So we know that the corals shuffle the different types of symbionts, and because we've seen colonies with different types of symbionts, some of these particle recognition could be assisting the corals in that process. And then, so here we have a similar graph, but these are focusing on immune and defense-related proteins. But we again see the same pattern, where we see over higher number of expressed transcripts related to immune and defense response. And again, I have a complete list there, and that just shows you that you can find in the paper if you're interested. That's how hard that work is of trying to explain when writing omics papers into how these molecules work and how they may affect the coral. But I picked one to highlight. A very interesting molecules is the membrane attack perforin proteins. And then you have a picture or a diagram of these. These basically are proteins that the coral can produce and then assemble and perforate bacterial cells. So imagine that membrane being a bacterial cell membrane, and the coral can produce proteins that will lyse the bacteria, and it kind of forms like a defense mechanism. And this particular coral species, I think, has 12 of these membrane attack complexes, and 7 of them were overexpressed in urban corals. And of course, this is a response to the high number of bacteria in the inshore waters. And another set of transcripts, these are that were very interesting, were the digestive enzymes. These were also in much higher number overexpressed in urban corals in comparison to the offshore corals. And these we thinking assisting again is because we're thinking that the Port of Miami corals are relying more on heterotrophic feeding than the offshore corals. many of these different enzymes have not been directly linked to coral digestion because there's not many studies of corals in the digestive process. But one, the serine peptidases, which were 14 of these that were upregulated, these have been functionally shown to exist in coral digestive system, and actually definitely are, helping to digest the food for the urban corals. These would be very good, I think, biomarkers for heterotrophic feeding for corals that could be developed into that. And the final group that I wanted to talk to you about were the transporters, and here what was interesting about different transport in and out of the cell is that usually you get even number of overexpressed and underexpressed, and because you have to look at the function of the transporters. Are they transporting exporting, or are they importing, or the transport is within a cell from one organelle to other, or the transport comes from the symbiont itself to the host. So it's a much more complicated story. And one of the few that I wanted to maybe highlight, if we still have time, is the ABC transporters in the C4 family. That are really responsible for foreign substance removal. So we're thinking, hypothesizing that they help the urban corals to remove potential substances that are toxic, like maybe pharmaceuticals or other toxins produced by phytoplankton, natural toxins. And the other group of overexpressed transporters are responsible for actually can be found in oocytes. So we're thinking this lipovitamin and intelligin are related possibly to these urban corals being able to reproduce, because these are proteins that are found in oocytes during oocyte formation. And very quickly to the conclusions, I just want to conclude everything that we talked about. Summarize the molecular mechanisms that allow Port of Miami corals to persist in highly urbanized waters, include urban coral host stress-resistant Durasinian symbionts that are better at photosynthesizing in shallow and turbid waters. High number of overexpressed pattern recognition receptors suggest that urban corals can differentiate between food particles and potentially pathogenic organisms. Similarly, the higher number of overexpressed digestive enzymes suggest that the urban corals depend on heterotrophic feeding for additional energy source. And then, on the other hand, the same enzymes might be used to protect the corals from potential pathogens because they enter the same route and maybe just digest it. Overexpressed defense and innate immunity proteins in urban corals contribute to their resistance against foreign microorganisms. And finally, altered expression of transmembrane and vacuolar transporters reflects coral acclimation to water conditions at their location. And with that, I just wanted to thank everyone at NOAA, AONL, and CMA's Coral Research Program, and the NOAA Omics Initiative for funding this study and funding my salary to conduct this study. And I will take any questions.
Thank you so much, Evelina. We'll now take any questions from our audience. Feel free to type them into the general chat. You can also ask questions from Anderson's talk as well if you didn't get a chance to after that presentation. So just wait a minute, see if there are questions coming in. Thanks, Tracy. Yeah, if you all could just take a moment to answer the poll question if you're interested. Will help us determine if this kind of information is useful to you all, or is this something that you'd like to learn more about? Elena, I had a question. I guess more like I'm curious about something. Could you just like speculate or talk a little bit about the, if any, potential coral restoration implications from this kind of research?
So I think our next step, we're still going to be researching these urban corals because they are very interesting, and one potential study that we are proposing and trying to get money for is to test if these corals would be good seed for restoration. Do they have to evaluate them for potential? Because Port of Miami is many times expanding, there's new structures being built or renovated, and many times we have corals that have to be relocated. We don't know yet if they are completely adapted yet to that shallow water, or can they be moved? And that's something that we want to test for sure next, because we definitely could use them, specifically since they have to be moved for restoration. So definitely we're thinking of it, and hopefully we will conduct some studies to address this.
That is awesome. Thank you so much. Yeah, it just seems like there could be a lot of cool restoration implications from these corals that seem to be resistant or resilient to a lot of those harsher environmental conditions. Thank you. Do we have other questions from the audience? We have a few minutes left. Please feel free to type them into the chat. So we have a question from Tracy. This is for both our presenters. Can you describe other possible uses of your work? I think we'll start with Evelina first, if you'd like to answer that.
Yes, I think so. Our work at NOAA is, I would describe it a transitional in nature, right? Because we are coming from academia where we study basic biological questions, and we want to know how the transcriptome or proteome responds. And we don't think of, I mean, it's not purely applied. So we're thinking of ways, how can we do the work and use it? And one way that I can think of is just as Anderson presented, to develop biomarkers. For example, some of those very strong genes that are overexpressed at certain physiological state of the coral, like heterotrophy, could be a marker where you can just go and see how the corals are receiving their energy. We know, of course, already of doing the symbiont typing and things. So I think it can be definitely developed into many different biomarkers of resistance. In addition, if you make their transcriptomic response and you have a gene, if you can sequence the genome, you can many times link the expression response to the actual site of change in genetics, which is called either single nucleotide polymorphisms or other deletions. They're many times connected, and then that becomes a genetic marker where you can just go and say this is marker for heat resistance. And some studies have been done like that by Palombi in American Samoa. So if you're interested in that, how you can develop markers for health, that's one person that is doing it already, and we just would have to like catch up with our study to get to that point.
Thank you. And Anderson, I don't know if you wanted to touch on that also before we wrap up. Don't think we're hearing anything from you, Anderson.
How about that? It's weird.
Yeah.
It seems like I have to hit it twice. Anyway, I think some of Tracy's examples she put in the chat of things where this approach would make sense, marine mammals, which I think is actually being done to some extent. There is some biomarker profiling work with marine mammals, seabirds, fish. I actually think those projects would bear fruit faster, because what I totally glossed over, and one of the reasons why proteomics is difficult for corals, is because they're these complex communities that Evelina kind of touched on. They have different symbionts. There's also different bacteria and things, and that makes doing the actual proteomic data analysis very challenging. So I basically started doing this with one of the most challenging organisms. If you were to do proteomics of fish or marine mammals, yes, they're going to have microbes and other stuff, but the majority of what you're going to be getting out is going to be from your target organism. So I think those people that would want to use this kind of omics health monitoring with non-coral marine organisms, you actually would probably get better data, and you could really hit the ground running, more so than you would with a coral, which is just more complex. So I think, that's a good way to look at, these data sets are a little bit smaller, but I think some of the issues that I encountered might be a little bit more coral specific. So I think proteomics, you're only going to see better looking data if you were to apply this approach with seabirds, mammals, and things like that. So I would say I'm glad that was brought up, and I definitely would encourage people to consider that approach in their research. I think you're going to probably see even better looking data than for coral.
Excellent. Thank you so much, Anderson. Really appreciate it. So we're a little past time. I'm going to wrap everything up now. Just wanted to say thank you so much to our speakers for presenting on a really interesting work today. We really appreciate you taking some time to do this. And thank you so much for all of our participants for tuning in and listening. Just some reminders, the NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time. Our next seminar will take place on September 15th, and is titled Environmental DNA Provides Quantitative and Efficient Estimates of Hake Abundance and Distribution in Support of Fisheries Management, which will be presented by Dr. Oli Shelton, a research ecologist with NOAA's Northwest Fisheries Science Center. So with that, we will end the webinar. Thanks, everyone. Have a good one.